All right, let's just get straight into it. You've probably spent hours drilling plant cell structures, right? You think you've got the basics down, but what if I told you there's this one tiny sneaky misunderstanding that is literally robbing Cambridge Biology 9,700 students of crucial marks every single year? Well, welcome to today's Explainer. We are tackling the 1.2 Large Permanent Vacuole and Tonoplast Exam Masterclass. And listen, we're not just rehashing what these structures are. We are diving into exactly how the examiners demand you write about them.
Now, check out this absolute classic student answer. Water moves into the tonoplast because it needs to dilute the large permanent vacuole. I mean, if you're just skimming, you might think, hey, yeah, that sounds like science. But to a Cambridge examiner, oof, this is the ultimate cold open mistake. It is a complete disaster.
So here's the million-dollar question for you. Why does that exact phrasing score 0 marks? Seriously, 0. Take a really close look at the terminology there. What is fundamentally broken about the biology in that sentence? If you can't spot the errors instantly, your grade boundary is actively at risk. But hey, don't panic. By the end of this explainer, you're going to be dissecting statements like that with the ruthless precision of a principal examiner.
And that brings us to why we're so hyper-focused on this exact topic today. 15%. That is roughly the chunk of total marks in your paper 2 that can heavily rely on how well you've mastered the large permanent vacuole, the tonoplast, and all those associated transport and water potential concepts. So yeah, this isn't just some minor footnote at the back of the syllabus. It is a foundational pillar for sure.
Which brings us to Section 1, Unlocking the Assessment Framework and the Method Mark Strategy.
Now, I'm going to reveal the exact examiner clue that flips your answer from a 0 to a hero in just a second. But first, you need the actual framework. Look, top-tier students don't just memorize textbook facts. They use this 4-step method mark strategy to show a deep connected understanding that works even in weird, unfamiliar contexts. Step 1, identify the core concept. Step 2, pull the evidence or data straight from the stimulus material. Step 3, apply your scientific reasoning. And Step 4, honestly, this is where most people drop the ball. Wrap the whole thing in exact examiner wording. Examiners give marks for this systematic pipeline. If you follow it, you're practically forcing the mark scheme to give you credit.
And if you don't believe me, listen to this direct warning straight from a Cambridge Principal Examiner report. Candidates consistently confuse key terminology, always define terms before using them in extended responses. This right here is exactly why ordinary revision just doesn't cut it. You might vaguely get how water moves around a plant cell, but if your baseline terms aren't explicitly correctly defined, the examiner literally stops reading and stops awarding marks.
So let's lock that baseline definition down right now. The tonoplast is the partially permeable membrane surrounding the large permanent vacuole in a plant cell. Look at the precision there. It's not just some layer, it is definitely not a wall, it is a partially permeable membrane. You've got to memorize this exact phrasing. It's the absolute bare minimum required to satisfy the examiner before you can even think about jumping into complex water potential analysis.
All right, moving on to Section 2, correcting the top 3 mistakes and evaluating evidence like examiners.
We're going to pivot to the specific procedural errors that examiners are constantly complaining about in their reports. If you can dodge these, you immediately separate yourself from the average candidate. Here are the top 3 traps you've got to actively avoid. Number 1, confusing cause and effect. That is a massive red flag in biology. Number 2, completely forgetting to check your units and terminology, especially when you're dealing with data. And number 3, giving these vague, generic answers that aren't specific to the actual experimental context they gave you. Drill this checklist into your brain. Every time you finish a practice question, run your answer through it.
And actually, this perfectly illustrates that first huge mistake. On the one hand, we have the phrase, vacuole gets bigger, so water enters. See how that totally scrambles cause and effect? It makes it sound like the vacuole is just magically expanding and sucking water in. No way. Now look at the top end phrasing. Water enters down a water potential gradient, increasing vacuole volume. Do you see the difference there? The scientific reasoning pipeline is spot on. The gradient is what causes the movement, and that movement is what causes the increase in volume. Boom! That's how you lock in those high-tier marks.
Next up is Section 3, Step-by-Step Question Methodology and Executing with Clear Working.
Okay, time to introduce our standardized sequence for processing really data-heavy stimulus materials. You know, when you flip the page and see a terrifying, massive experiment about plant tissue, here is exactly how you deconstruct it. It's a 5-step sequence. 1. Identify the key elements, like are we talking about the tonoplast, water potential, or solute concentration? 2. Extract the data from whatever tables or graphs they throw at you. 3. Execute your analysis using really clear notation. 4. Verify your results, like do they actually make biological sense? And 5, present your final answer using that precise, correct format and terminology. Honestly, this framework is completely non-negotiable if you want to crush unfamiliar contexts.
All right, let's actually put this into practice. Check out this mock comparative data we have for plant cell A and plant cell B, detailing vacuole volume alongside tonoplast permeability. I want you to mentally pause for a second here and predict. Extract that data. What's the relationship? If an examiner asked you to compare these two cells based purely on this data, how exactly would you structure that answer? This is the moment where your skills are actually put to the test.
Because if you just mindlessly listed the numbers, you've completely missed the mark. Top-scoring candidates show real critical analysis by hitting these exact criteria. You absolutely must identify similarities and differences. You've got to use comparative connectives, words like whereas or however. You have to back up literally every single claim with extracted evidence straight from that data. And finally, you have to draw a justified conclusion about how tonoplast permeability actually relates to vacuole volume. That is what evaluating like an expert actually looks like.
Which brings us to Section 4, the diagnostic cliffhanger and the hidden cause of lost marks.
Look, mastering the biology of the vacuole and the tonoplast, nailing the definitions, understanding the gradients, crushing comparative tables, that's honestly just step 1. Because the actual command words used in the exam prompt dictate your entire mark band. Take a look at this question. You know the biological concept now, for sure, but do you genuinely know the difference in how you need to write your answer if the examiner asks you to describe the tonoplast function versus if they ask you to evaluate the data? It's wild, but a single hidden command word changes the entire required response. I mean, you could write a flawless description, but if it's an evaluation question, you hard capture marks no matter how perfect your biology is. So what does this all actually mean for your final grade?
It means that single word is the hidden trap costing top-tier students their A stars. So, how do you decode these command words so you always hit that top mark band? Well, that is exactly what we are going to solve in our next explainer. Make sure you subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCS, so you can excel in your Cambridge examinations. You really don't want to miss that next masterclass where we decode the exact command words that dictate your grade. See you there!